thing will turn off, you mean? Great. So, hello, and it's a great pleasure for me to be able to give you, to talk to you today about stem cells. This, I have to confess, is a unique experience for me, actually giving a lecture where I can't see the audience, where I'm actually talking into a camera, can't see the response. But this is probably a unique experience for you too. Let's give this a shot. So for the next hour, you can see the title. I'm going to be talking about stem cells, but I'm going to be giving it kind of a bit of a unique spin, maybe a little bit different than you're used to hearing about it. What I'm going to do is remind you throughout the entire talk that even if you're interested in the translational value of stem cells, what you really are developmental biologists, and that stem cells are fundamentally part of developmental biology. And what you'll also see is that this guides, this kind of bias, will even permeate the way that I think about using stem cells in cell-based therapies. So this also leads into an aspect that probably is on the mind of many people in the audience, which is, I enjoy the science, I love the biology, but when can I actually use this to treat a patient? And this has kind of been in the background of the stem cell field from the earliest days of the stem cell field. Even before we understood the complexities, and it's the complexities I'm going to spend the hour talking about. When is it appropriate to attempt a therapy in the face of imperfect and incomplete knowledge, recognizing that we will never have complete knowledge and that our knowledge will always be dynamic and changing. It will always be imperfect. These are kind of my guiding principles, and they've been my guiding principles from the very beginning of the field. When do you try something new? And stem cells and regenerative medicine are something new. And I guess I would say is, of course, when standard of care is not optimal, of course when it's safe, of course when what you do is not going to deprive a patient of better proven options. But probably what we have here is when what you want to do makes absolute biologic sense. Does it make sense in terms of our known biology of the cell and our known biology of the disease, where the gaps exist? Recognizing that these are constantly moving targets. So when I talk, this is going to be guiding a lot of the way I present some of these data, which means that if you want to put this into biotech terms, that one needs to know the mechanism of action. That becomes very important. That's a guiding principle. One also has to be very secure in the proof of concept. So I'm going to be spending a lot of time talking about that from the vantage point of the stem cell. And then, of course, clinical competitiveness means that what you want to offer still is better and safer, and ideally maybe even less expensive and less harmful and less painful and less invasive than what already exists out there. So the themes that I'm going to cover over the hour are the following. Even if you lose contact and you don't listen to another word that I say, these, I think, will be the important take-home messages that I want to leave with you. And that's to reiterate the way I started. Stem cells are the building blocks of the body and of each organ of the body. They are ultimately part of fundamental developmental programs. Their job is to put an organ or a system together, that's organogenesis, and then to maintain that organ's function all throughout life, even in the face of perturbations. That's why nature puts stem cells there, and that is what we need to understand. When we want to use stem cells therapeutically, what we have to realize is that we're harnessing these programs and trying to invoke them or re-invoke them. We can't go at cross purposes to what their biology is meant to be, but it does open up all kinds of possibilities. Think of it as rebooting the computer or reseating the line. Now, for the cells to do this, they engage in an enormous amount of crosstalk and complexity. We tend to think of stem cells as purely these cells that are going to do cell replacement, kind of swap them in, swap them out the way you change a carburetor in a car. But cell replacement is just one of the mechanisms by which nature puts stem cells there to re-achieve balance in a disequilibrated system. They do many, many other things. They change the milieu, they chaperone cells, they restore normal cellular metabolism. And in some respects, that may actually be lower hanging fruit, more attainable than cell replacement. More tractable, and in the end, maybe even more important. Not that we're not going to get to cell replacement, but as you'll see, that we may be able to make an impact taking advantage of what may even be more important parts of the fundamental biology. And I won't have time to get to this, but of course, a whole area of regenerative medicine is the notion of taking the stem cells, growing them in a dish, and discovering what the mechanism, what the drug target might be for a particular disease, and then, of course, finding the drug that may actually change that. So it's not the cell that goes into the patient, but it's the drug discovered from the cell that goes into the patient. I won't have time to get into that, but I think that's an important aspect to try to remember. Now, how did I personally get into the stem cell field myself? Well, I think this is also going to be important for any clinicians in the audience. I started getting into the stem cell field before it was called the stem cell field. And this is an example of what we like to call not bench to bedside, but bedside to bench. This is a picture of a CT scan of a baby that I took care of. I'm a pediatrician, and particularly a pediatric neurologist and neonatologist. And longer ago than I would care to admit, I took care of this baby who had a stroke. The baby went on to get completely better. And I went to my professors and my attendings, and I said, how could this possibly be? There was no MRI. These are before the days of MRIs. And I said, but how could this possibly be? How could a baby have a stroke like this, and then after seeing the baby at about a year of age, the baby was completely better? And they said, well, kids do that kind of stuff. But I wondered, how did they do that? What was the essence of this kind of plasticity? Because if we could understand it, then maybe we could harness and do it in situations where this does not happen. So we went back to the bench, from the bedside to the bench, and found that contrary to what had been the case, the prevailing dogma for the nervous system and for other parts of the body, the left-hand model, a very deterministic model, we found that there were actually cells, like the yellow cell on the right, that could give rise to many different cell types much later in development than you would ever imagine. Well, these yellow cells are cells that came to be called stem cells. And they started opening up the whole notion that the body was more plastic than we ever imagined, including rigid organs like the nervous system, that cells existed that perhaps could be extracted, expanded, and then put back, that would recapitulate this kind of natural kind of plasticity. So those were cells before the advent of the stem cell field. Now, as you know, we do have a very vibrant stem cell field. And I would like to say that even though sometimes the political debate forces us to pigeonhole various kinds of stem cells, that's not actually the way nature works. That, in fact, there's a continuum from cells, let's say, that exist in the inner cell mass, that we now call embryonic stem cells to cells in the fetus that are now going to give rise to all the various cells of the body and into adulthood. Now, the political debate has forced us to talk about embryonic stem cells and adult stem cells. But what all of us are really doing are focusing on what the cells are doing right here during development. If you're starting on the extreme left with the blastocyst, you're trying to teach your cell that it's meant to be living in the fetus, giving rise to the equipoise. If you're on the extreme right, you're trying to fool your cell into thinking that it's living in the fetus, giving rise to these various organ systems. And it's these kind of signals that we want to recapitulate. Now, we also learn that this continuity, this continuum, not only goes from left to right, but now also goes from right to left, because we now have the ability to take cells at the extreme right and do various kinds of reprogramming. It simply, and it can be your favorite way of doing this, you can take a cell and push it back into developmental time so it acts as if it's still living in the blastocyst and still can give rise to the various kinds of cell types that you see here. Now, these, of course, are called induced pluripotent stem cells. Now, these kind of cells, and I guess you can't see the movies, but they can give rise to all of these various kinds of cell types. And you can get, trying to play this now, beating cardiomyocytes. That would be slide, I guess it would be slide number 1, or video number 1, Tracy. You can sort of see beating heart cells that one can actually do a kind of a stem cell EKG with. Blood vessels. maybe I won't use so many videos. It looks like it slows things up a little bit. It would be video number 2. Okay. So what I want to do, do we have the slide down again? Okay. So before we start talking about using the cells for transplantation, I do want to make a bit of a nod to the fact that this is still, the importance of stem cells still is most profound in teaching us about embryogenesis, organogenesis, and then the various homeostasis preserving mechanisms that exist in the stem cell that, again, we want to harness. So, again, in this lecture, I'll be talking about the top step, what we're going to be doing in vivo. Perhaps, if I don't screw up too badly, I'll be invited back to give a second lecture where later on I can talk about using the stem cells in vitro to model diseases and model processes. However, even if we want to talk about in vivo, I think it's important to understand what the logic is, what the program is that the stem cell is starting to carry out. And well, let's go back here. As you can see, let's talk about one of the very first programs, and that's this inner cell mass, or the epiblast deciding that it wants to first give rise to a nervous system. And this was a question that Ilya Singech in the lab wanted to do. He wanted to see whether, in fact, he could actually start getting neural tube closure in a dish, the formation of the neural tube and the closure, the earliest stages of neurogenesis. And in fact, he was able to do that, as you can see here, able to get very nice neural tubes in a dish. But he was able to do it by recapitulating development. And he was able to find that if he used the right formula that the epiblast actually uses to give neural tube, he could very, very quickly get pure neural stem cells in a dish, particularly if he used small molecules to close off all the other fate options, prevent them from giving rise to endoderm or mesoderm, or even staying pluripotent. So he could give rise to early neural stem cells and then cascade his way to getting the various kinds of more mature neural stem cells. This is an example of pure neurons. And the default pathway, interestingly, is to give rise to neurons that are interneurons, GABAergic or glutamatergic, living in the dorsal forebrain and the cortex. Now, that doesn't mean that one can't give rise to other kinds of cells, but there you actually need to give them very, very specific instruction. Tell them you need to be ventral, so you give it sonic hedgehog. You need to be in the ventral mesencephalon, so you give it FGF8, or you inhibit GSP3 beta if you want to get dopaminergic, or if you want to get motor neurons. Now, once you have this, there is this notion of being able to get stem cell, to get these cells, particularly if you took these cells from a patient who had a disease. You could have cortical neurons, for example, that have autism, or that have Rett syndrome, or that have Alzheimer's disease. Now, this is not a new notion. In the early days, we were able to go to diseased fetuses after miscarriages, or even to postmortem tissue in adults, pull out the neural stem cells and say, well, we have a neural stem cell that has Alzheimer's disease. So this notion of modeling diseases in a dish is not new. With the advent of the embryonic stem cell field, we could go to infertility clinics where they were doing pre-implantation genetic diagnosis, and there would be a blastocyst that had a disease that could not be implanted, that would be discarded. Well, now we could make embryonic stem cells, turn them into neurons just the way I showed you, but these would be neurons that had otherwise a lethal disease for us to study. But again, what we would love to do is to be able to get to cells that do not involve the death of the donor, even for those kind of cells that are formally kind of inaccessible. And that's where these induced pluripotent stem cells, these iPS cells, became particularly useful, being able to get difficult to obtain cells from a living patient. Now, I extolled the virtue of getting pure stem cells, pure lineages in a dish. Now, everybody could say, and again, this is going to come up when we're talking about trying to repair the nervous system, that that's not really the way the nervous system is built. In fact, that's not the way any organ, the vertebrate body plan is built. It's not just pure cells, it's pure lineage. It's lineages interacting with each other. So every medical student will tell you that every organ, every limb, every extremity is vein, artery, nerve. The vasculature comes from the mesoderm. The nervous system comes from the epiderm. But nevertheless, you get the embryonic organ, and then you get the fetal organ, the neonatal organ, the adult organ, and everything is completely coordinated. Well, that must be another kind of system where lineages crosstalk to each other so that there's constant co-patterning. And that was something that Jeff Lindquist and Seth Acevedo in our lab, in collaboration with David Cherish, they were joint postdocs between the two of us, wanted to explore. And I won't play this video again, but you could see that we could get vasculature very, very nicely. But at the exact same time as we were getting vasculature, we also were able to get nervous system. And I would like to play this video, if you could. I guess this video would be video number 3. I'm not sure. I guess you have to play that, Tracy. Is that, oh, could you keep playing it? I think it's on a loop. Is the video playing? All right, well, this is a minor, we better go on. What you would see is that the two lineages just follow each other like trains on a tram track. They just completely co-pattern each other, vasculature and neural. That's a program. And that if you, the interesting thing is what initially sets the neurovascular pattern is not the central nervous system, not coming from the neural tube, but actually coming from the neural crest. Once that happens, and what you can see here is the blue cells are smooth muscle, the green cells are neural crest becoming autonomic nervous system, the red cells are vascular endothelial cells. And they together emerge simultaneously, crosstalk to each other, and if you get rid of any one of them, the differentiation, the emergence of patterns of all of them completely fall apart. It's an obligate crosstalk between them. After that pattern is set, and I guess we need to go to the next video, please. I guess this will be video number 4, but maybe that's, you can see that then, oh, okay. All right, why don't we just skip it? Some of the other videos that we'll have to play for longer. But the central nervous system in blue then comes out and joins the parade, then joins a pattern that had previously been established between the neural crest and the autonomic nervous system. But it's an obligate crosstalk that needs to take place. So let's say, however, this is all arcane stuff, and you really don't care so much about this fundamental biology. What you really want to do is be this guy drilling into a head of somebody. Just let's get cut to the chase and let's start fixing things. I would make the argument that even then, what you are is a translational developmental biologist. And what you need to know is to be able to harness these programs. And it really doesn't matter. Let's use the nervous system as an example and the neural stem cell as an example. It doesn't really matter how you get to become a neural stem cell, whether you want to do it from ES cells, or iPS cells, or fetal tissue, or even from the adult. You're going to get a neural stem cell, and it's still going to be the same fundamental biology that you need to understand. And that is this notion that when you take a stem cell and you put it into a pathologic host, there's a dynamic, and both elements change. The stem cell changes, the host changes, and it changes in response to these various arrows. And it's these various arrows that one needs to begin to understand and to identify. So let's start understanding what these arrows are. Sorry. That the arrows will play out fundamental developmental programs that are inherently in place to compensate for these perturbations. So what would be the first arrow that one would look at? Let's look at this very top arrow, as you can see over here. It's the notion that the host tells the neural stem cell a come hither kind of signal. If there's pathology, what we were able to observe is that the stem cell seemed to gravitate to where there was pathology. Now, what was interesting is that it did it in a whole series of kinds of pathologic situations. And initially, this was very perturbing. It happened in ischemia, in trauma, in tumors, neurodegeneration. Now, all of these had a different etiology, a different tempo. So we had to figure out what was it that they all had in common. The common denominator to all of them was an inflammatory signature. Now, we had always been taught that inflammation is bad, and it is bad. But it's kind of Janus-based. It also has another side to it. And in fact, that's what we call this paper, inflammation's other side. And what we realized is that it was the inflammation that the stem cell was keying into. And the prototype that we used at the time was SDF1 alpha as the inflammatory chemokine and CXCR4 as the prototypical receptor. What this shows is that after an infarct on this top, there was a huge recrudescence of SDF1 alpha, and that's what the human neural stem cells would home to. On the intact side, not at all. And the cells would do it in a one-to-one correspondence. What was expressing the SDF1 alpha was the active astrocytes and inflamed vasculature. So it made an enormous amount of sense. Now, if we wanted to harness that, we couldn't just take the pure SDF1 alpha. Because you really would actually create more harm than good. But what you could do is optimize the binding and minimize the signaling. And that's what we've done to actually make a peptide based on SDF1 alpha that exacerbates and that allows homing to come under our control. And I'll get to that in another way. Now, what else could the host be telling the stem cell? It may also be telling them that of all the things you can become, we want you to become this particular cell type. And that's driven by the host. So here are all the various cell types that one might see. Oligodendrocytes, neurons, and astrocytes. And we wondered if we went to a situation where neurons were missing, would the stem cell shift to become that missing kind of neuron? Now, the answer is complex. So the answer was yes. But it wasn't wrong. It was just naive. What Jeff Matlis and I did was go to a very artificial situation, taking advantage of a very clever model that Jeff developed, in which he was able to, as you can see over here, one small part of the adult mammalian cortex, that one little part of the brain where there shouldn't be neurogenesis, he developed a method of targeting apoptosis of pyramidal neurons. If we took the neural stem cells and just in that one particular box, put them there, not outside the box, there they became glia cells, which was very appropriate. But inside the box, they would give rise to pyramidal neurons. And some of these pyramidal neurons would actually send processes across to the other side, as you can see from this track trace. Well, that seemed cool, and it established the situation that where there's injury, even in the adult, one can start seeing new neurogenesis. The cues kind of get recapitulated. Where it was naive was the fact that it was a very artificial situation, and that most diseases aren't like that. Well, that was neurons. We wondered, could we do the exact same thing with glial cells? So we went to a situation where glial cells were actually going to be degenerated. There was a mouse, and I guess we will need the movie for this, called the Shiver Mouse. Could you play this movie, please? the amount of shiver is dramatically reduced. And we'll go back to the slides. And I think you can see this is that panel C is the amount of shiver, the whole body tremor of an animal not treated. D is the amount of tremor that was eliminated. It happened because the oligodendrocytes were becoming the dysfunctional, replacing in that particular case, the dysfunctional oligodendrocytes. And we've been able to now go to some other models. The blue here represents cells in a mouse model of a leukodystrophy in a kid, Krabbe's disease, called the Twitcher mouse, where you can see that the blue cells have distributed themselves throughout the neural axis, and where there normally are no oligodendrocytes are now giving rise to oligodendrocytes. These blue represent a whole halo of host axons that are being myelinated. As you can see over here, this is all donor-derived myelin with very nice major densities there. Now, both of these models, they were encouraging in the mid-90s, but it was naive. The targeted apoptotic model doesn't recapitulate itself in nature, that kind of pure apoptosis. The shiver mouse has no correlate in human disease. Most diseases are very complex. It's not a single cell. It's not a single pathologic process. And we learned that lesson when we started actually going to a stroke-like lesion. As you can see, that and this is a study that was done by Coop and Park, a neonatologist who wanted to look at a stroke-like lesion in the rodent. The arrowhead above points to very devastating lesion from hypoxic ischemic injury. What Park did is he took the cells, as you can see, on the bottom frame, and put them down, into the left, and they would move from their implantation in the left all the way over, migrating across commissures and corpus callosum to the place where the infarct was, all throughout the cerebrum. If you put the cells directly in there, as you can see in this next slide, they completely filled out, and there's a higher power view shown towards the right. If he looked at what the cells were becoming there, he saw, as you can see in the other panel, that they became, they certainly became neurons. I'm going to see whether I can do any kind of pointing. Well, I guess I can't. Let's forget the pointing. Okay. So they became neurons that also send processes across to the other side, but he saw something else that was also exceedingly interesting. He saw that they were becoming oligodendrocytes, as you can see in that left panel. And if he started doing the math, he was able to show not only were the cells becoming new neurons, but they were becoming new oligodendrocytes, new astrocytes, even new neural stem cells. In other words, what the stem cell was doing was trying to recapitulate the entire environment. And often what I liked, it knew that we didn't just need neurons. That's not what ischemia is. It knew it needed to give rise to neurons, but it also needed to give rise to the oligodendrocytes and to the astrocytes. And I often like to say that even the dumbest stem cell is smarter than the smartest neurobiologist. It knew that this is what we need to recapitulate, and it had programs that would start doing that. So there had to be a degree of crosstalk between these cells for this kind of thing to happen. And the crosstalk was the stem cells talking to each other, giving rise to a division of labor. So this is a schematic of one of the ways that we were able to, that we've kind of teased out. This summarizes a whole lot of data. The neural stem cell is cycling. It looks as if it does give rise to either by stochastically or by instruction or by design, it gives rise to a neuron. The neuron then sends out signals to its host, to its sister and brother cells that even if they wanted to become a neuron, they are actively inhibited from becoming neurons. They become non-neurons or glial cells that then, and a lot of the signaling can be through BMPs, it can be through match delta, it can even be through codes in the neuronal membrane. But now the glial cell will feed back onto the neuron. The neuron grows a receptor to it. Now, GDNF is just one example that I'm going to be coming back to a lot of a whole series of these kind of factors that are chaperoned by the glial, that are chaperoned, that serve as a chaperone by the glial cells to the neurons. The neuron grows a receptor to it. Now, this makes an enormous amount of sense. It's a self-assembly mechanism where the non-neuronal cells coming from the neural stem cell, from the same clone, will chaperone and protect. Now, if you take that biology and you do a transplant into an abnormal host, well, then the host becomes the fortunate beneficiary, of this kind of process. Now, the factors are stimulus appropriate, they're quanta, they're pulsatile. But now the host has benefited from this. This is called the chaperone effect. It's mediated by the PA3 kinase pathway. And it's so important because I would venture to say, with very few exceptions, every therapeutic benefit to date that we have seen has actually not been through cell replacement, but through the chaperone effect. And in fact, even if you replace neurons, it's a good thing to also replace, or at least have them chaperoned by the glial cells. So once that happens, because of that crosstalk, now you have the stem cell talking back to the host and bathing the host in a whole series of various diffusible factors. Now, if you take the stem cell and you stick it into the ventricles are lined by a germinal zone. The germinal zone is the home of the stem cells, and you place them in there, and they will distribute themselves throughout the brain. Turn the slide too quickly. But they will move out there because that's really what they're normally meant to do, distribute themselves throughout the brain, integrate in a seamless fashion in the substance of the brain, and one could start imagining that, what about if this was something, not just a gene that gave a color, but something that you cared about, that could actually bring in a bioactive molecule in a Trojan horse fashion. What kind of diseases would this be pertinent to? Well, it would be pertinent to this whole class of lysosomal storage diseases. These rare diseases, but in the aggregate, fairly common and affect the pediatric group because all of these kids miss within their brains an enzyme, which is a normal housekeeping gene for the stem cell, that now, if the stem cell is distributed throughout the brain, just cranks it out and cross-corrects these kind of cells, almost like a bone marrow transplant to the brain. So lysosomal enzymes could be one of the things, and in fact, the use of stem cells for these diseases back in the early 90s turned out, as you can see in this paper, to be the very first therapeutic use of a solid organ non-hematopoietic stem cell. Again, tapping into its normal fundamental biology. And Jean-Pierre Lee did this kind of paper. This is now some later work, but you can see in the upper left-hand corner, the cells distributed themselves throughout the brain, and as a result of restoring, in this particular case, hexosaminidase B, could forestall the onset of disease, preserve motor function, and significantly prolong life. Not because it was replacing cells, but because it replaced, through its normal housekeeping function, missing enzymes. The cells also have a direct anti-inflammatory effect. Here's an example of the cells actually diminishing directly activated microgliosis, or actively inhibiting the amount of inflammatory cytokines. And what we're starting to learn is every disease, every neurodegenerative disease, every injury really has an inflammatory signature to it. And this simply summarizes the whole host of mechanisms by which the neural stem cells, other than cell replacement, though there was some degree of it, were able to kind of complement and make an impact on this type of neurodegenerative disease. The cells also have a direct neuroprotective action, anti-scarring, mobilize endogenous stem cells, and make those cells promote neurite outgrowth, grow, have even an effect on the host vasculature. Now, here's an example of where all of that kind of observations come together a little bit. Because I don't want to slow things down, I'll just describe this and not use the arrow. But if you look at the middle, the middle panel, you can see all kinds of green. That's scarring. It's GFAP. But it looks as if the scarring comes to a halt after this infarction in something that almost looks like the map of Greenland. Well, if you like to do jigsaw puzzles, you can look up a little bit, and you can see on the top panel, that red area, those are human neural stem cells, and it almost looks as if that would fit into where the scarring is stopped. And the merged image on the bottom shows, in fact, that. Where the neural stem cells are, the scarring stops. And now what used to be formerly a no-man's zone, a no-man's land, is an area that allows now, as you can see in the bottom panel on the right, allows host cells to send out processes that can navigate through an area that formerly was excluded from them, simply because the scarring had been inhibited. What's another action? Well, if you look at the bottom panel, you can see the blue arrowhead is pointing to fibers crossing the cortex, which are completely fine. There's nothing wrong with that whatsoever. That's exactly the way it should be. But if you go to the top panel, and you can see this is an area of the brain in the extreme left-hand part that was infarcted. But it was infarcted and now invested with neural stem cells. And you can see that now the fibers that normally should just be, kind of perpendicular, now are rather giving rise to these long, delicate, arcing fibers that are bending and starting to send fibers into this area that was damaged, but now trying to join the parade, participating, as you can see in this next slide, in part of the neural network. And not only doing that, but inviting this new neural network, this new parenchyma, to be vascularized, as you can see in panel D, with the arrow is pointing to that. Now, formally now, taking all of these processes together, as you can see in A, an area that would have enormous amount of parenchymal loss, now seems to have a reconstitution of that parenchyma. Now, is this beautiful textbook parenchyma? Absolutely not. In fact, it's a bit of a mess. It doesn't have this beautiful lamination. But is it saying that the stem cell and the host are starting to talk to each other in a way that tries to reconstruct the milieu? I think that is what it says. And even though it wasn't pretty, this animal actually did better. So again, tapping into a fundamental process. We learned this from hypoxic ischemic injury, from stroke-like lesions. Can we apply it to spinal cord injury? Well, Ted Tang decided that he was going to look at this particular process, and he did a whole huge hemi-resection. And what he wanted to do was see whether he could take stem cells, simply put it on a scaffold, and bridge this huge gap. And what I want to do is show you the data first, the video. Could you bring that up, please, Tracy? This becomes important. This actually is a very important video. And we're going to show you data that first Well, this we found to be really pretty exciting. Let's move to the next. And of course we thought, well, this is great. Let's book a plane to Stockholm. Stem cells are the greatest thing since canned beer. Do I need to move this to get to the next slide? So we figured we need to figure out what was going on. Well, we simply looked at the histology and we saw all this green staining. This is great. These are neural fibers going through the epicenter. When we stained to see who was giving rise to the neural fibers, that's where the surprise came. The red cells are our cells. They are not the ones giving rise to the neural fibers. In fact, all these neural fibers bridging the injury were coming from the host, not the stem cells. But yet, they were having the impact that you saw, which again made us recognize the power. In fact, it helped us understand what the chaperone effect actually was. What the stem cells were doing, here again, is the epicenter circled here. The green represents the scarring. Where the stem cells are, the scarring stopped. And now it was the host fibers that were allowed to be able to now go through the center. It's not pretty, it's a little bit ratty, but it was sufficient with both motor and sensory, sufficient to help restore some of the function. Now, here you can see the red cells, those are the human neural stem cells. The green cells are host neurons. The green cells are not becoming the neurons, but now, because they are expressing a whole host of trophic factors, the animal's own neural fibers are coursing through them. If they're there, as you can see in this MRI, the syrinx seems to be reduced in size. Now, does that mean that we can't get a little bit of extension points? For example, we know that this has to be multifaceted, but can we get little bridges from the neurons, from the neural stem cells, as illustrated by this arrow? Well, I think we can, but in a muted fashion. But it can also help the situation. These are examples of donor-derived bridging neurons, as we can see them. Now, are those playing any kind of role? Well, here's the spinal cord of a rat, and if they are playing any kind of a role, if we put a track tracer down below, as you can see over here, track blue, where that arrow is, it should go up the axon and turn that cell blue if it's really sending a fiber down there. And in fact, that seems to be what happens. So some of the cells, not all of the cells, as you can see there, the red cell, that's a donor-derived cell. It shows that it's a human neuron. And of those 3 cells, that one's blue because it has sent a fiber down there. So we are getting some cells that are bridging the gap like that. Now, what they're doing is bridging the gap, and what we think they're doing is invoking intraspinal circuitry. So another way of invoking circuitry that can mediate some of movement that don't involve higher centers. But if you take the protection, you take the anti-scarring and the pro-angiogenesis, and invoking these various kinds of intraspinal segments, taking it together, now you start getting some improved function. The kind of function that I was able to show you before over there. Here, this is motor evoked potentials. It's not beautiful, it's not perfect. Roman numeral 1 shows motor evoked potentials, Roman numeral 2 shows sensory evoked potentials. Each one, A is what you would normally see, C is what happens without any kind of intervention, B is after intervention. So it's not normal, but it looks to be significantly better than what you would see in C. And the BBB scale seems to be better. It's a combination of the stem cell being the orchestra leader of trying to mediate all the various kinds of actions that are going to be necessary for spinal cord injury. What we're able to show, in fact, is that the same kind of thing can be extended to even large animals, to large primates. Same kind of process, the cells and the scaffold shown over there, diminishing the amount of neurodegeneration, as you can see with the silver stain on the left side, decreasing the amount of inflammation, as you can see on the right side. So that is the scaffold and the stem cells, with the asterisk showing decreased degeneration, and on the right side with decreased inflammation. So in the interest of time, I'm not going to go through this. I think you'll be able to access the list of all the various kinds of actions, the multiple mechanisms that are required that underlie recovery following spinal cord injury mediated by the stem cell. And you're going to start seeing that I'm recapitulating this theme over and over again. In Parkinson's disease, same type of thing. Homeostatic effects, multimodal from human neural stem cells. Multiple actions. Parkinson's disease, as you can see there, can be modeled by lesioning a monkey with NPTP. The neural stem cells there are implanted into the nigra, into the striatum. And what you could see is that in the top graph, that the amount of Parkinsonian symptoms were reduced compared to a sham control. Not to normal, but dramatically reduced. Now, how did that happen? Again, through multiple mechanisms. Some of the cells did become dopaminergic neurons, as you can see here, but not enough of them to account for recovery of function. Maybe only about 3%. So it was there, but that's not what was responsible for the recovery. What was responsible for the recovery was preservation of the host nigrostriatal pathway. The red cells here are the donor cells. They're not becoming the Th positive cells. Those are the animal's own Th positive cells that normally would have died that are not. And now, what we can see in this slide is that the donor cells are right next, cheek by jowl, with the host Th positive cells, making GDNF, causing diminished inflammation, preserving the pathway. These are examples of rescue host dopaminergic neurons, looking kind of nice. Here's an example of a preserved nigrostriatal pathway. These are the functions, so it wasn't recreating this pathway, but it was preserving the pathway that existed there, as you can see. And then, for some other reason, for a mechanism that we actually do not understand, one can start seeing that there was diminished alpha-synuclein aggregates. Again, we don't know the mechanism, but the stem cell was somehow also restoring normal metabolism to the cell, allowing this kind of thing to happen. So again, multiple mechanisms, which now brings us to a very recent result, to ALS. Now, it used to be thought that ALS was a simple disease. We're starting to know now that it's not a simple disease. When we started looking at this disease, we thought it was going to be just like the experiment that Jeff Mackless and I did. ALS is motor neurons that die. The experiment that Jeff and I did with pyramidal neurons just being knocked out should be the same, just moving down to the spinal cord. Well, what we learned is, as many of you in the audience know, ALS is exceedingly complex. It's many different kinds of pathologies, and you're not meant to look at this except that this is Don Cleveland's way of saying, look how many things are going on. So now we wondered, in fact, could the various mechanisms that we're starting to learn the stem cell was playing out be matched to the many pathologic mechanisms that are playing out in ALS. And the fact that we were able to publish this paper about a year ago shows that that seemed to work. So here is where we do need, hopefully you can play this video. Are you able to play this video, Tracy? not only should he not be able to do that, he actually should not even be alive. He's walking the road a lot at over 200 days of age, and most of these ALS mice, the SSOD1 mouse, die at 90 to 120 days. But yet, as a result of the intervention, and the intervention was taking the neural stem cells and going into multiple cardinal areas throughout the neural axis. Delayed disease onset, slow disease progression, improved performance. But now, why was it able to do that? Well, here's an example of how we transplanted the animal. And here's an example of not only a delay in motor function, but as you can see in this Kaplan-Meier curves, that a subset of the animals, about a quarter of the animals, reliably had very prolonged survival, some to almost a year and a half. Why was that happening? Well, if you look in panel B, you can see some of the cells did become motor neurons, but that was not what was doing it. Those motor neurons that we saw were not mature. They were not sending processes out through the ventral horn. They did not have mature markers. So even though we saw some of them, those could not be responsible for what we were seeing. But inevitably, as you look at higher power, you can see that the few cells that were becoming motor neurons, even immature motor neurons, were surrounded by a huge number of cells that were clearly not neurons. So we started looking at those. And those cells that were not becoming neurons, but surrounding both our donor-derived neurons and the host motor neuron, were cranking out a huge amount of GDNF. And as a result, as you can see in N, they were preserving the host motor neurons beyond the critical threshold of 10%. And as you can see in O, the motor neurons that were preserved were making good, intense, intact, functional neuromuscular junctions. P shows on the top that if we went to the cervical region, to the respiratory drive center, we were preserving their motor function and preserving their respiratory function. The patients, as well as the mice, die a respiratory death, and we were allowing respiratory function to be preserved. So early on, had we been trying to look for a release or a bioassay, we would have taken the neural stem cells, put it, let's say, in a spinal cord slice, seen that they were giving neurons, and we would have said, oh yeah, the cells are great. Now our release criteria is not. What we do is take a spinal cord slice, take the blue stem cells, put it out there, and we look and quantify neurite output. Because then we know, as you can see in the actual data and then in the schematic, in a quantifiable way, we are having cells that are going to effectively create neurotrophic factors that can do this. Now, as you can see in Roman numeral 1, it has to do this throughout the neural axis. Its action is not changing this genetic, the amount of mutations in this animal. So it's the manifestation, which suggests that even though we're using a mouse model that models familial ALS, maybe if we're getting to the output, to the functional manifestation, this can also be applied to sporadic ALS. What's another mechanism? Now, if you look in 4, B shows a lot of astrogliosis. A in Roman numeral 4 shows a diminished amount. Initially, we thought, well, what we're just doing is just decreasing some inflammation. Not that that's trivial, but we actually, as we started learning more about ALS, we started also realizing that we're having another very powerful mechanism. Anybody who studied ALS now recognizes, and this is shown in this graph in the upper bars, that astrocytes are one of the toxic mediators in ALS. And what we saw, if we looked at the natural history in this mouse, was that curve in red was rampant astrogliosis. And the more rampant the astrogliosis, the more virulent was the disease process. What we were doing, in fact, was having another action. with our exogenous stem cells, we were changing the fate of the host neural stem cells and inhibiting the host neural stem cells from making what we know are toxic astrocytes. And it's the astrocytes that are mediating some of the cell death. We were inhibiting the host neural stem cells from making toxic astrocytes. We're also then replacing them with our own gentler, kinder, more protective astrocytes. And we're also giving rise to protective gray matter oligodendrocytes. So another mechanism. Then, as you can see in Roman numeral 5, we were diminishing inflammation, activated microgliosis. Of the motor neurons that we were preserving, we were diminishing the amount of neurofibrillary tangles, as you can see over there. So again, multiple mechanisms, and in the interest of time, I won't list them, but the stem cell, with almost a one-to-one correspondence, its multiple mechanisms against the multiple mechanisms that you are seeing as a manifestation of ALS. Now, this becomes a regulatory nightmare, because now you've got to give a cell that you want migratory, that does many things, that can give rise to many different kinds of cell types. But that's the biology that we need to harness, and the regulatory agencies are going to have to come and recognize that. Now, it's not just diffusible factors. There's also some cell-cell. There's another mechanism that we discovered again by accident, and that's that the stem cell on the host will actually make cell-cell contact, establish gap junctions, and all of you know that gap junctions are, and that's what we illustrated here, again, a surprise, the gap junctions are these pores through which molecules of a particular size can move. But not just molecules. Also, it can be nucleotides, it can be microRNA, it can be many different things. We don't know what's going through there, but we know that it's important. Again, we discovered this by accident. This is a mouse mutant that has an abnormal cerebellum. In the interest of time, I'm not going to go through what's wrong with the cerebellum, except that this animal, called the nervous animal, loses its Purkinje neurons. The Purkinje layer, as you can see in the top, is this beautiful, it almost looks like a pearl necklace of this one cell. Well, in this animal, the pearl necklace, as you can see in the bottom panel, seems to disappear by early adulthood. If you take a neural stem cell and you implant it after the disease has manifested itself, nothing happens. You don't change the course at all. However, if you implant the stem cell on the day of birth before the disease manifests itself, so there's another lesson there as to how we use cell therapy, now you see something interesting. The pearl necklace exists. The Purkinje cell layer is preserved. If we look to see what was constituting this Purkinje neuron, and I think you know where I'm going with this, it was not in any way, oops, I guess that slide didn't play. Oh, wait a minute. That slide didn't come up. Oh, that's too bad. What you would have seen, and I guess I can show it over here, is that the stem cell here in green is completely surrounding the host Purkinje neuron, making cell-cell contact, decorating it with these white dots, which represent the formation of gap junctions. If you block the formation of gap junctions, you completely lose that rescue effect. That happens in the nervous animal. Now I'm showing you an example of it happening in other kinds of Purkinje cell degeneration. This is a really different mechanism. The only thing it has in common is Purkinje cells dying, and this is spinal cerebellar atrophy type 1. Now, what are the gap junctions doing? We don't know. In this very rare mutant, what it was doing is an interesting kind of gene therapy. That animal is characterized, as you can see in this parabola, by having too high a level of TPA. Not TPA the way you know it for stroke, but TPA as the upstream effector of signal transduction. And what the stem cell somehow is doing is pushing it back, as you can see in this arrow, to the sweet spot. where viability of the Purkinje neuron is at its peak, the peak of the parabola. So it's a different kind of gene therapy, which is also another take-home lesson, not putting in a gene, but re-regulating the animal's own genetic function. Maybe that's what the gap junctions are for, re-regulating intracellular metabolism. So here's the schematic here. The stem cell actually influencing the amount of expression of TPA, and then the downstream effectors are improved. Better neurotrophic factor handling, and better mitochondrial function. But it's not just, and then making the whole thing more balanced. And it's not just in these kind of inherited rare diseases. This is an example of exactly the same thing happening via gap junctions in spinal cord injury. So what are the many things now that the stem cell is doing? And summarized here, crosstalk and the chaperone effect, which means that the stem cells are not through cell replacement, but engaging in the crosstalk that is going to completely reorder the milieu. Now, can we actually change the endogenous stem cells? Well, there's an example of doing that, and again, there's both a positive and a negative lesson that's acquired from that. And again, as you can see, a lot of what we're learning are the kind of lessons that you learn by either being really lucky or making a mistake and then reinterpreting. So the notion would be, could we use the stem cell to deliver a transgene that's going to affect the host stem cell? So the stem cell here is just a delivery. And I'll get to that in a second. Now, we wondered, NT3 is known to take stem cells and make them more neurons. Could we get more neurons? So we thought, well, let's make the neural stem cell express lots of NT3. And what would happen is it would spill out into the milieu. It would feed back onto the neural stem cell's own receptor, so we get more neurons there, but then also feed forward onto the host and get more neurons there. So we did that. We did that in that same stroke-like lesion. It's a little complex for me to do without a pointer, but the red are the stem cells, the blue are all the neurons, and what we simply showed is that we got lots and lots of neurons. So the donor cells became many, many kinds of neurons, and that's what all those orange and yellow cells are. Donor red cells staining for the green new end, becoming lots and lots of neurons. Many more than just the normal amount of 3 to 5%, 80%. And I'll come back to whether that was a smart thing at all. And they went on and they had the right kind of neurotransmitters. But what the arrow shows is not only did we get more donor-derived neurons, but in the oval you can see more host-derived neurons too. The NT3 working on the host neural stem cells. Now, initially we thought that this was great. And it was. But do you really want 80% neurons? That's too many. The right ratio is only about 3 to 5%. Now, this animal did okay, did not have seizures, but I think we dodged a bullet here, and it wasn't so smart. Another example of where it wasn't so smart is illustrated over here. We went into the spinal cord with exactly those same kind of cells. Remember I mentioned in the ALS model that the stem cell, the native stem cell, putting it in there, gives us lots of motor neurons. If we put in the neural stem cell that was now making more NT3, we started getting fewer motor neurons, fewer chat-positive cells. Now, it wasn't that we were getting fewer neurons. We were getting lots of neurons, but we were getting the wrong kind of neurons. Now, what shows over here, we were getting lots of sensory neurons. Sensory neurons could underlie allodynia, or pain. This is not what we want. So what we were doing, in fact, is playing with one system, trying to give us more neurons with NT3, was making us, as you can see, lose all of our GDNF, which was doing something equally as good. So it taught us a lesson that playing with one pathway can have unintended consequences on another beneficial pathway, which says that sometimes it's not smart to muck around with Mother Nature. So, is there a situation, however, where you could put in a transgene? And that, in fact, is illustrated over here. That there are situations where you may want to deliver a stem cell, and all you want it to do is to deliver a transgene in a global fashion. And that's what brain tumors. Brain tumors are very migratory. They elude the best surgeon, gene therapist, radiotherapist that exists, because the cells break off and create these other satellites. That's why brain tumors are so incurable. Well, we wondered, if we armed the stem cell with a tumor-killing gene, it could find those satellites, deliver the tumor-killing gene, and eradicate them. And there, we didn't care what the stem cell became. It didn't even have to exist and persist, as long as it didn't cause any mischief. Those experiments were something that Karen Abudi, who was a postdoc in the lab, started exploring, taking advantage of this pathotropism to deliver. In fact, it works. We use GBM, or tumors, as our example of showing that this exact happens even in the adult brain. The intact adult brain, very little migration. The injured or abnormal adult brain, different situation. The whole terrain is different, and that's what she showed. Brain tumors were the example. This has now been replicated by many other labs using cancers, and is in fact giving rise to an FDA-approved clinical trial now using stem cells to deliver oncocytal genes to glioblastoma, but taking advantage of a fundamental program of the stem cell, which is a pathotropism, or in this case we would call an oncotropism. As illustrated over here, you can image the stem cell, even if it's on the opposite side of the tumor, homing in on the tumor, that if you stick it into the tumor, it will go to the edge of the tumor and stop right at the interface of the tumor, as you can see over here, stop right at the interface of the tumor and the normal tissue, except where a stem cell is moving in and invading normal tissue. And it'll take the tumor and diminish it to about 80% of the size. You can stick the stem cell even into the vascular system, and if you have metastatic cancer to the brain, it will home to the brain. Here's an example of metastatic breast cancer. Again, the stem cell, only in the situation where there's metastasis of the brain finding, as you can see here. So here are the potential actions of a stem cell in neurologic conditions. I know I'm going over my time. I'm almost finished, and I'm going to try to wind up with some important take-home lessons that I think are important. These are the mechanisms that I think the stem cell, and you can see most of them are inherent to stem cell biology and do not involve neurostem cell replacement. Will we get to neurostem cell replacement? Yes, we will, as we will get to heart replacement and all that, but we're just not there yet. So, but that does not mean that we're incapacitated in taking advantage of its biology for translational reasons. So when do we actually get here? Well, we're starting to get there. There are some clinical trials, mostly taking advantage of the chaperone. The biggest obstacle to date so far has been, how do we exploit stem cell biology? And I'm listing here all the things that we do not know. But the biggest thing we do not know for most diseases is what actually needs fixing. So we can have the best stem cell that exists, but if we don't know what it is that needs to be fixed, we don't know what to deliver or when to deliver it. So those who study the disease have to make progress hand in hand with rework in the stem cell. The other major challenge to the field is how to deal with chronic injury. Everything that I or any other stem cell biologist has shown you to date involves acute, fresh spinal cord injury or any kind of CNS injury. But most of our patients have chronic CNS injury. They've been in wheelchairs for years, which means that And all the biology that I've been telling you really happens in an acute environment. So that's illustrated over here. Oh, sorry. I guess under the parabola is where all the activity is. But not only is that window of opportunity lost when you get to the chronic injury, but also whole other things have interposed themselves. There's atrophy, and scarring, and disruption of connections and vascular supply. So it's going to take a whole other paradigm, not just using pre-existing strategies. And that's illustrated over here that if you take the exact same stem cell and you expose it to an injured environment in a dish, and the only difference is whether the tissue that's damaged was acutely injured or chronically injured, and then you do a differential display, what you can see using the stem cell as a reporter cell, that it sees different things, it acts in different ways. With the acute injury, there's an upregulation of genes that mediate migration and proliferation and differentiation and anti-apoptosis, like the accelerator. If you expose it to tissue that was injured in the remote past, it downregulates all of those genes and upregulates genes that inhibit migration, inhibit proliferation, inhibit differentiation, are pro-apoptotic. So the cell sees something different. So maybe one thing is to try to recreate in the chronic injury the window of opportunity that existed with the acute injury. That's one mechanism. But again, it's going to take whole different kinds of paradigms, as you can see over here. Different paradigms where you actually do real reconstruction. And in the interest of time, I won't be able to go through that, but this is the same stuff that I showed you earlier on about our spinal cord injury, where we actually use biomaterials to create other ways of getting through the spinal cord, to the muscle, around the injury, reconstructing circuitry. And here is an example of stem cells on biomaterials and scaffold, and you require them together. The lesson is that it's always going to be better to protect what you have than trying to recreate the kind of complexity that exists as a result of normal development. What are the best cells to use? Well, this makes for a huge, almost Talmudic debate in the stem cell circuits. Here are various ways, for example, to get to the neural stem cell. They all have their pros and their cons. As you can see here, those on the right locked in to the neuroectoderm. You're not going to get tumors, you're not going to get inappropriate cell types, but they don't proliferate as much. The cells on the left, very proliferative, easy to scale up, but they need to be tamed. The chromatin's not as compacted, and you can get tumors, and you can get inappropriate cell types, as you can see. And here's a paper from Steve Goldman where in the Parkinson's animal, even after he got good therapeutic outcome from TH-positive cells after the ES cells, he didn't get teratomas. It wasn't poorly differentiated cells that he also saw. He started seeing primitive neuroectodermal tumor. So neuroectoderm that just wasn't mature enough. So that's, if you want to use non-neural cells, and there's a lot of people in the audience right now who want to think about doing that in the heart or the nervous system, just remember, these are not going to give rise to heart cells, they're not going to give rise to neural cells. You're doing it because you're trying to harness their molecular role. But always remember that the stem cell is still going to follow its biological imperative. You may be taking it out of its environment and putting it somewhere else. It does not know that. And it is still going to follow its biologic programs. And here's an example of where this investigator, in this bottom paper, took mesenchymal stem cells and put it into a model of multiple sclerosis, which is a very inflammatory disease. The mesenchymal stem cell simply saw the inflammation, and it started to proliferate. and it just made a mass. It was just following its normal imperative. So remember, one of the cautions of putting a cell where it doesn't normally belong is understand what its biology is. So you may need to pick cells based on the disease, based on the action, based on the biologic program that will play out its normal biologic program in both a safe and effective way. You may need to do this empirically, but the goal is always going to be that you want to find something that restores homeostasis. And this simply kind of recapitulates all of the actions that I've shown you today. So circumspection does not mean a retreat from combat. Cautioning you to be circumspect does not mean, and this is actually a quote from me, does not mean that this is not going to be promising. It means that one needs to be careful. So in conclusion, and I know I've gone over, I apologize for that. I guess I didn't know the technology as well as I wanted to. Here are the various summaries that you should take away. That ultimately, stem cells are models of developmental programs. That re-invoking these developmental programs, particularly those that are homeostasis promoting that underlie plasticity and adaptation, are the ones you want to re-invoke, whether it's exogenous or endogenous. Multiple mechanisms, multiple cell types engaging crosstalk is what we need to be cognizant of and re-invoke. It's better to work in concert with these programs than working at cross purposes to them or bending the program to be the way you think it should be. Try to understand what it is and then harness it in an intelligent, rational kind of way. Molecular therapies are going to be the low-hanging fruit, more so than trying to get the particular cell type that you want. We're still involved in a huge amount of intellectual heavy lifting to get there, which means looking how the cell type got to be there to begin with, plus even trying to understand what cell type you want to replace. Schizophrenia, autism, even ALS. There we were mistaken. We thought we just wanted to replace motor neurons. We were wrong. We wanted to replace the astrocytes. Disease in the dish technology, that's also the low-hanging fruit. The drug goes into the patient. Then recognizing that inflammation has a Janus face. There's a love-hate relationship between inflammation and the stem cell. The stem cell homes to where there's pathology, then it becomes anti-inflammatory, but inflammation also does harm. Remember the pediatric age group. I think I showed you that time and time and time again. We started off understanding the stem cell biology by looking at models of pediatric disease and moving up to the adult. So it wasn't trickle down, it was bubble up. Because stem cells ultimately are developmental biology. The kids are developmental biology playing themselves out. And that chronic established injury is still the major, major challenge for regenerative medicine. We are nowhere near to approaching the patients who, in fact, are our largest constituency. So these are the people that I need to thank, and I'll be glad to take any questions with whatever time is available. Sorry I went over, but I'm happy to entertain any kinds of questions. Thanks. So I guess, do you talk to me or do I just look for the Q&A? I guess I'll look for the Q&A. Oh, okay. So, okay. Okay, that's great. So question number one is, does it work for Alzheimer's disease? The answer is, we don't know. There are people that are trying Alzheimer's disease, trying it with Alzheimer's disease, based not on replacing cell types, because we don't know what kind of cell type to replace. But if Alzheimer's is inflammatory, maybe we can restore inflammation, damp down the inflammation. If there's certain trophic factors that might be useful, like BDNF or NGF, either taking the amount of neurotrophic factors that are intrinsically made by the neurostem cell or getting the neurostem cell to make more of it is also being tried. It is not being tried in patients yet. It has had in animal models has not really had the effect that we'd like. The next question is, does the death cell program still work with these new cells? They die if they don't make connections. They die if they're crowded out. However, if there's a niche, they will integrate. And they seem, it's very interesting. I'll add something to your question. Host cells died because of inflammation or oxidative stress or toxins. It's interesting that the neural stem cell, maybe because they're young, seem to be more resistant to metabolic toxins, oxidative stress, to excitotoxicity. So they actually can survive even when some of the host cells, maybe because the host cells have a genetic flaw, do die. What happens with, the next question is, what happens with the blood-brain barrier for brain tumors? Well, the blood-brain barrier seems to be compromised by brain tumors anyway, and that may be one of the mechanisms by which the stem cell can make it through the vascular compartment into the pathology. But it also does it through another mechanism. The stem cell does seem to be able to make it through the blood-brain barrier by squeezing through by various kinds of mechanisms, probably some of the integrins and things like that. Also, it seems to just have its own biology that allows it to go through in a way that hematopoietic cells do not. What is the impact of the pH in your experiment? We've not looked at that. I'm wondering whether you're talking about these STAP cells, these cells that become reprogrammed because of a very acidic environment. We do not see an effect in pH other than to kill the cells. That whole area of acidic reprogramming, as you might know, is coming under debate, and it's unclear whether it's a real phenomenon. Can stem cells repair damage made to peripheral nerves by chemotherapy on individuals? Actually, there are a lot of people investigating that, and the quick early answer is we think that they can, but it needs to be explored more. It still doesn't do it well enough. Can stem cells help with pulmonary fibrosis? That's a hypothesis that's being explored. In fact, there was just a paper that came out in the pediatric literature trying to use mesenchymal stem cells to inhibit the scarring that you see in the lung disease that premature babies develop. That looked a bit promising. Now, that may happen just through conditioned medium. It may happen through the anti-inflammatory, anti-scarring effects of the stem cells. It's a very attractive hypothesis. We think it might work to some extent. Things never work as cleanly as you'd like, but that is a hope. Well, the next question is, well, the rest are not questions. Oh, okay. In my opinion, which reprogramming strategies would be more suitable for clinical translation? I would go with non-integrating technique. For right now, I would use episomal reprogramming mini loops that do not involve a virus, do not involve integration. Things may change if the small molecules become better, if the proteins become better, if the microRNAs become better. Maybe that will change my mind. Right now, and we would love to participate in transplanting iPS cells. The technique we're going with, because it seems to be safest, most efficient, cheapest, easiest, and most scalable, is episomal reprogramming. So that's where we're going right now. On this particular day, this particular year, this particular talk, I'm going to go with that. I think I've run through all the questions. I can take some more. I think I have about a minute left, or maybe I have no time. Okay, I guess if there's no other questions, should I? I think, all right, I'm supposed to say that's a wrap. So I apologize for some of the technical snafus. I apologize for going over. Thank you very, very much for listening and for being so patient with me. Hopefully I'll be invited back to give you the in vitro disease in addition. Thank you. Bye-bye. What do I do now, Tracy?